Hi, good morning. Our lesson for today is cell division. So our first topic is mitosis. So what is mitosis? Mitosis includes the replication of DNA material so that the daughter cells will have the same genetic information. So it is a process by which two new daughter cells are generated having the same number of chromosomes. That means it is diploid as the parent cell. So our symbol is 2N. This includes the division of the nucleus to produce two nuclei. So in mitosis, when the cell splits into two new cells, and both of these new cells have the same number of chromosomes as the original cell, 2N. Mitosis is a way of the cell to make two new cells, and these two new cells have the same chromosomes. So that means what? The new cells are also diploid. Diploid. What's the meaning of diploid? It means that they have full set of chromosomes, just like the parent cells. This process also involves the nucleus of the dividing cell into two separate nuclei. So we have here the illustration of the phases or the stages of mitosis. Okay, it is a continuous process. From the cell cycle, interphase. After interphase, we proceed to the mitosis, and it involves a prophase, a prometaphase, metaphase, anaphase, telophase, and cytokinesis. Most of the time, it is just prophase, metaphase, anaphase, telophase. Then, the division is the cytokinesis. First, we have to review about the interphase. So, the cell cycle, most of the time, the cell undergoes cell cycle. The interphase. The interphase is divided into gap 1, synthesis, and gap 2 before you will proceed with the mitosis. So what happens in the interphase as a review? The interphase happens before mitosis. It is like a cell's growth and preparation phase. So during the late interphase or the gap 2, the cell has made a lot of new stuff and there are now two chromosomes or centrosomes in the center cells. So there are two centrosomes here in the center cells. Inside the cell's control center, and that is the nucleus, the genetic material or the chromosomes has copied itself, but it still looks like tangled mess and can't be seen as individual chromosomes yet. So it's like the cell is getting ready for the next phase. So interphase is just getting ready for the next phase. Okay, so we will proceed with the first stage or the first phase of mitosis, and that is the prophase. This is the initial step, the first step of the mitosis or cell division. In prophase, two important things happen. What is that? What happened during this prophase? The first thing that happened is that the inside of the cell control system or the nucleus, look at the change of the nucleus. The messy genetic material or the chromatin gets organized. It's organized and become visible as distinct chromosomes. So, makita na nimo na organized na siya. Makita na nga distinct chromosomes na siya. These chromosomes look like pairs of sticks stuck together. So that is the first thing that happened. Next, in the cell center, tiny structure called centrosomes, look at the changes in the two centrosomes, start moving apart. Diba sa interface, tapad sila, karon they are moving apart. And little tubes, or the microtubules, begin to form, which will help in dividing the cell later on. Okay, so these are the spindle fibers. So they are moving away from each other, the centrosomes. Okay, so in prophase, when the cell gets its act together, it organizes its genetic material into clear chromosome and starts setting up the tools it needs for cell division. Next, we have prometaphase. So look at the nucleus. Look at the chromosomes. So in the prometaphase, a protective covering around the cell's nucleus, which is the nuclear envelope, breaks apart and disappears. So we don't see the nuclear envelope anymore. It disappears. So the microtubules, or the spindle fibers, which come from the ends of the cell or the pose, reach out towards the center where the genetic material or the chromosomes is. So na-separate na siya ang mga centrosomes. So, naa na siya diri sa each pole from north and then south. And then, the spindle microtubules are helping, no? This one. The chromosomes to line up towards the center. It reach out. The spindle microtubules reach out towards the center where the genetic material or the chromosomes is. Each chromosome has a special spot called kinetochore that looks like a black dot. This is the kinetochore, the black dot. Some of these microtubules grab into these kinetochores, making the chromosomes move around a lot. Other microtubules from the opposite ends of the cells also connect. Special proteins help in pulling the chromosomes toward the middle of the cell. So in this stage, the goal is to line up the chromosomes towards the center of the cell. So hindi pa siya nasa center. Going to the center. Next is the what? Okay, review. So during this metaphase, the cell gets rid of its protective covering and tiny tubes starts grabbing and shuffling the chromosomes around, getting them ready for the big split. So in the metaphase, this is where the chromosomes line up in the middle. So metaphase is like a balancing act during the cell division. The cell has set up a structure called mitotic spindle, which is like a scaffold with poles at each end. So these are the poles. So the spindle, mitotic spindle, marghilo. Okay, so the chromosomes, which look like pairs of sticks gathered in the middle of the cell on an imaginary line called the metaphase plate. It's like they're lining up in the center. The middle part of each chromosome, called the centromere, is all lined up in its metaphase plate. Each chromosome has its two sticks the sister chromatids. They are connected in the pulse ends of the spindle by tiny tubes. It's as if these chromosomes or each chromosomes is being held by thread from both sides. So na thread from both sides. Therefore, during metaphase, the chromosomes get into a line in the middle of the cell. They're on a tightrope, which their centrosomes are all lined up, ready for the next part of the cell division. So, it's lined up. Next, we have the anaphase. So, the anaphase, when the cell splits its genetic material into two. So, look at the separation between the two sister chromatids. So, one sister chromatid, this one splits into two. So, the other one is going to the North Pole, and then its half is going to the South Pole. So, in anaphase, each chromosome, diba, it is a pair of sticks held together by the centromere. This one, the black one is the centromere. So, look at the difference. The centromere is here, two centromere, then the two sister chromatids. In this anaphase, the one separated becomes two. Okay? So, in anaphase, this centromere splits up, separating the two sticks, which are the sister chromatids. So, the sister chromatids splits up. They split its centromere. Special motor proteins in the cell, powered by ATP, help move these separated sister chromatids towards the opposite ends of the cell along tiny microtubules. It's like they're being pulled apart to opposite sides of the cell. So, they are pulled apart. Nagbulag na dyan sila. Okay? So, while this is happening, the tubes or the microtubules connected to the separate chromatids get shorter, and the ones not connected to the chromosomes get longer. This action pushes the pole or the end of the cell farther apart, making the cell stretch out. So, by the end of the anaphase, each end of the cell has an equal share of chromosomes. So, in anaphase, this stage is when the cell divides its genetic material in half, with one set of chromosomes going to each end of the poles, going to the North Pole and going to the South Pole, or whatever is the direction, as long as they are going in the separate ends of the cell. And it stretches out to make room for the two new cells. Okay? And after anaphase is the telophase. It is the last act of cell division. What happens in the telophase? Okay, the cell elongation started. Under the microscope, you can see that this is the telophase. The cell keeps stretching out as it did in the anaphase, making it longer. So, at the ends of the cell, new control centers or new nuclei starts to form around the chromosome. So, the nuclei starts to form. You can visibly see under the microscope that it is turning into a nucleus. It's like the cell is building little houses for the genetic material. So, the way chromosomes and the structures that help in cell division, the spindle fiber, look in telophase is almost the opposite of how it looked in prophase. Things are getting back to normal. So, you can see it is forming a nucleus. So, by the end of the telophase, the genetic material or the chromosomes relax and unwinds, and the cell division tools the spindle fibers disappear. So look at the spindle fibers. It is disappearing. Okay, so in telophase, it is when the cell finishes stretching, builds new control centers around the genetic material, and the things start looking like they did before. The whole cell division process began. So this means the cell division, the mitosis is done, and there are now two new cells with the genetic information. So after the telophase, the cell proceeds into the cytokinesis, the division of the cytoplasm. So what happens in the cytokinesis? It is the final step of cell division. It usually happens at the same time as telophase. So as telophase finishes up, the cell is almost divided into two, with each end having its nucleus and a set of genetic material, which is the chromosomes. So in cytokinesis, it is the cell physically divides into two separate cells. So in animals, it is like the cell pinches itself in the middle to achieve separation. Okay? So in this final step, the cytokinesis of the cell division process, it results to two new daughter cells. So try to look at the cytokinesis of the animal cell. It is like pinching to form two cells. Okay? So while both animal and plant cells undergo cytokinesis to divide to create daughter cells, they employ different mechanisms due to their distinct structural characteristics. So in animals, it uses contractile ring to pinch the cell membrane, while in plants, a contractile membrane na, this is the contractile membrane, to pinch the cell to form two new daughter cells. While in plants, what happened in plants? Okay, plant cells form a cell plate to create a new cell wall. Cell plate. This is the cell plate. So in animal cell, we have contractile ring, while in plant cell, we have cell plate. Okay? So these differences reflect the unique characteristics posed by the presence of a cell wall in plants and the absence of cell wall in animal cells. So in animal cells, cytokinesis involves a process of cleavage. The cleavage furrow. Okay? The cell membrane pinches inward to divide the cell into two daughter cells. No new cell wall is formed, while in plant cells, cytokinesis involves the formation of the cell plate. So vesicles bring material to the center of the cell, forming a new cell wall. This cell wall eventually divides the cell into two daughter cells. Okay, this cell wall. To form new cell wall, we will use the cell plate. Okay? So that is the end of the mitosis. So why mitosis is important? We have various reasons for it. Okay? So mitosis is important for various reasons, and it plays a crucial role in several essential biological processes. First, mitosis is important for growth. It allows organisms to grow by increasing the number of cells in their body. New cells produced through mitosis can divide and specialize, contributing to the development of the tissues, the organs, and the entire organism. How about cell replacement and repair? So mitosis is essential for replacing the damaged or the dead cells in the body. So when you are injured, or when the cells are injured or it died, mitosis ensures that identical, healthy replacement cells are generated to maintain the tissue function and integrity. And also, for asexual reproduction, In many single-celled organisms and some multicellular ones, mitosis enables asexual reproduction. Through mitotic cell division, an organism can produce genetically identified offspring, ensuring efficient and rapid reproduction. Tissue generation. In certain animals and tissues, mitosis play a vital role in the tissue generation. So, regeneration, to regenerate the tissue. What is the example for this? So, it is like in the animals, like you can see the starfish, or the salamanders, mga butiki. So, mitosis allows them to regrow the lost body parts such as their limbs or their organs for regeneration. Next, we have development. So, during the development of multicellular organisms, the mitosis ensures that the organism grows and forms into their correct body structures. It allows for the controlled increase in cell numbers at specific developmental stage. And the last is homeostasis. So, mitosis helps maintain the balance and stability of an organism's internal environment. And that is known as the homeostasis. By replacing old or damaged cells with new ones, mitosis contributes to the overall health and function of the organisms. Okay, and that is the end for mitosis.